Good morning, ladies and gentlemen. My name's David Williams, and I'm the Chairman of INOVIQ. My pleasure to welcome you here today to the 2024 Annual General Meeting. Our Company Secretary, Mark Edwards, informs we've got quorum, so I'll declare the meeting open. Right, that's me done on the formal presentation, formal proceedings of the AGM. I'd now like to hand over to Dr. Leanne Hinch, our CEO, to give you a formal presentation, which I said was already uploaded to the ASX this morning. So you can see it there, and people online can probably see it in front of them now. As I said, this will be videoed. All of this will be videoed, but this will be videoed in particular. And at the end of it, Leanne, if you don't mind, I'll come back and ask you a few questions about, just to sort of personalise it a bit more. Then I might go on to Greg, and then we'll take questions at the end of that. I'm sure none of us would care if you wanted to ask questions as we go, but if not, don't worry, I'll give you a chance right at the end. All right, Leanne.
Thanks, David. Welcome to everybody that's here today. I'll try and keep this presentation relatively short, but as David said, you're welcome to ask questions later. So INOVIQ is developing next generation diagnostics and therapeutics to improve outcomes in cancer. The majority of our pipeline is really powered by our exosome science. We have basically proprietary technologies, which means that they're in the process of being patented or are already patented. Two technologies, our exosome platform and our sub-B2M technology. We've got products in market, which include our ExoNet capture technology, which we've partnered with Promega, and also a bladder cancer test. And we have a deep pipeline across basically research tools, diagnostics, and therapeutics. So as Mary pointed out earlier, one of the really good things about this company is that we actually do have a deep pipeline. We have very good data across that pipeline, and coming up, we have a couple of announcements before the end of the year on some of our products. And our real focus is to partner for growth, whether that's to commercialise our products or in the later stages of development, particularly with therapeutic products down the track. We're well funded. We had $10 million at the end of September, and those that read the announcements would have seen that we recently got a $1 million R&D tax incentive return based on our R&D work, which was a lot more than that, by the way. And our key shareholders, obviously David Williams is one of our key shareholders. We've also got Merchant Funds Management and Biotech Capital Management. So the company's vision is to be a leading exosome diagnostics and therapeutics pipeline. That's really based on our exosome platform, which we consider very important to the company. Our focus, as everybody's aware, is on cancer. Why do we call our exosome technology a platform? The reason we call it a platform is we can put it in a box and sell it as a research tool, which we've done. We can create better diagnostics, or we call them more advanced diagnostics. They can be for screening purposes, for early detection of disease, or they can be for companion diagnostics, which is to select the right patients for future therapeutics. And that's really how we marry our exosome technology or exosome diagnostics with our future exosome therapeutics. And the reason that we moved into therapeutics is probably pretty obvious. It's a very high value area, and essentially we have leveraged the company's expertise, existing expertise and capabilities in that area to expand into these high value therapeutics. We also have our sub-B2M technology that's entirely focused on diagnostics, and those tests are for monitoring cancer, which is after people have been diagnosed with disease. We had a big financial year 24, probably bigger than shareholders appreciated in terms of some of our some of our milestones in general. One of our key milestones was securing our partnership with Promega for distribution, global distribution of our ExoNet products. We secured that in July. I'll actually go into that in a moment. On top of that, we've advanced obviously our diagnostics pipeline across our exosome ovarian cancer test, as well as our sub-B2M diagnostics as we're moving them through the development pipeline towards commercialisation. And we also progressed our early stage exosome therapeutics pipeline. We raised $9.4 million in order to fund that pipeline development and commercialisation activities. And we expanded our board, both obviously with David Williams at the end of last year and more recently with Mary Honey. This is really talking about clearly a lot of interest in our ExoNet technology now that it's commercialised, and our Promega partnership. Essentially, Promega, as I said, we secured the agreement essentially in April. They placed their first order in June. So they really started pushing this technology or our products into the market in August. Since August, they've actually secured 33 customers. So that's about 8 customers a month. They're pretty pleased with that progress. I'm sure, frankly, we'd love to see it grow faster, but it takes time with a new technology to get it into the market. What's important is that the applications that they're seeing or the interest they're seeing from customers includes cancer, it includes neurological diseases, there's actually a huge range of them, different cardiac conditions, transplant rejection, we weren't expecting that one, sepsis, and there's a variety of other applications. The type of customers that they deal with, we talk about up here. So they split the market into what they call academic and government, pharma and biotech, and clinical and hospital. What we're really trying to show there is the academic segment, it's much smaller in that they tend to do small research studies. So they don't use a lot of the product, and it takes a while for them to order product because they've got to have a project, they've got to have grant funding before they're ready to kick off. But they're really important customers to us because they validate the technology across a whole range of applications. So we only focus on cancer, but they do a whole lot of things. So it means that our technology can be used for all sorts of things. They validate it, they put that in publications, and then they do scientific presentations at conferences. And that means that pharmaceutical companies and diagnostic companies see those presentations from those key opinion leaders. So it's really important to us as we bring those customers on. And Promega, as you can see, they've got about 18 of those customers now, and they are focused on the key opinion leaders. The other segment they call pharma biotech. These are really important customers to us because these are going to be the large customers that drive sales. It takes time to bring these customers on. Couple of examples. If I talk about what does a diagnostic company look like, a diagnostic company could have already developed an exosome diagnostic. So it might be at what we call a laboratory developed test, but they haven't developed it using what we call a scalable technology. So we have a number of companies that are currently evaluating our technology because of its scalability, meaning that they can massively ramp up sales as their tests become successful on market. So we've got a number of those projects ongoing. Promega has brought many of those customers to the company, and so they've done a great job in working with them. The other area is pharmaceutical customers. So pharmaceutical customers, they're developing what we call immuno-oncology products to treat cancer. These products are expensive, and a lot of regulatory authorities now require that a companion diagnostic goes to market with these products. So there's a lot of interest in developing exosome-based diagnostics to select the right patient for their products. So that's a growing area. Again, takes time to find those customers, but they'll be very important customers to the company as we grow our sales. The other area is clinical and hospital. That's actually where we've actually got products in market or early stage projects that are being developed for diagnostics. Again, Promega's got a lot of clients in this area and generally for their product range and are slowly expanding ExoNet use with those customers. One of the key points that Promega makes is they expect to bring on a lot of new customers in first quarter next year as they've been speaking with these customers and they've got upcoming projects. So in terms of the outlook, we do expect to see sales grow from ExoNet. We've got new customers that are coming online. It will take time, but we expect this ExoNet business to continue to grow, particularly as we bring these new products online, like NeuroNet that we've done for capturing brain-derived exosomes. We've got a TexoNet product for capturing tumour-derived exosomes. And we also build what we call custom nets for customers that are developing specific types of diagnostics. Promega is actually working with us collaboratively on an ongoing basis to develop new products and combination products with their own products. That actually enables them to offer their customers a complete solution, whether that's for isolating exosomes and then RNA, or exosomes and, let's say, protein, or providing specific solutions for customers that are developing urine-based products. They're doing loads of work with us, so it's a very positive and collaborative relationship. So in terms of our exosome platform, exosomes are essentially small packages that are released by all the cells in our body, whether those cells are healthy or diseased. Those exosomes, which you can see there, those exosomes contain really important messages. We call those messages DNA, RNA, and proteins. We can actually capture those exosomes, read the messages inside to determine the health or disease status of the cell. That's essentially how we develop our diagnostics. Or we can actually put, let's say, as Greg says, a bomb inside that exosome, put an address label on the exosome, send it off to a certain cell type, i.e. it could be breast cancer, lung cancer, etc., to blow up the cancer. That's its job, and that's really what our exosome therapeutics pipeline does. So what we call our pan-exosome capture product, that's ExoNet, that's the product that's on market, that's partnered with ExoNet. We've proven it that it's faster than other technologies. It's very efficient at capturing exosomes. Basically, what it captures is very pure, rather than a whole lot of garbage that's contained in the blood that isn't exosomes. If you want to capture exosomes, clearly you want to just capture exosomes, we can do that. And most importantly, is our technology is scalable. What does it really mean? It means that we can take a very early stage experiment, let's call it from the bench, and we can take that all the way through to market as an actual diagnostic. And that's why we have diagnostic and pharmaceutical companies talking to us about our technology. It's because it is scalable. We have the technology to do that in our lab, so we can actually run thousands of samples in our own lab on behalf of other companies if they want us to do that. And we're expanding partnerships with what we call contract research organisations overseas, so they can offer those services to their actual clients, which are diagnostic and therapeutic companies. The company's published a whole lot of data on this. That's in peer-reviewed scientific journals, and we've got expanding publications that are coming out from various collaborators. As an example, a recent one was from a Spanish research group, and that was actually in multiple myeloma, again, not an area that we're in. And interestingly, their project was sponsored by Pfizer. So we're getting a lot of visibility with our technology now, which we're very excited about. More recently, as I think most investors are aware, we made available our product called NeuroNet. That's for capturing exosomes from the brain. Very excited by this product because essentially exosomes can cross what's called the blood-brain barrier, meaning it can carry out the messages that are in the brain. We can use that to develop earlier detection tests for we or others for Alzheimer's disease, Parkinson's disease, and various other diseases. What we've shown in terms of that graph on the right is that we can detect a whole lot of proteins that are different between people that have Alzheimer's disease and healthy patients. We show that some of those proteins are increased in Alzheimer's disease, some of them are decreased. Those are proteins, of them we found 47 that were unique to Alzheimer's disease. They could be used to develop, for example, a new therapeutic, or we could use them to develop, anyone could use them to develop a better diagnostic. So very excited by those results. We also validated the technology in Parkinson's disease. In terms of our own exosome diagnostic pipeline, we have an academic collaboration with University of Queensland to develop a screening test for ovarian cancer. I think you've heard us say many times there isn't a screening test available for ovarian cancer, meaning that it's diagnosed at a late stage, usually stage 3 and 4. which means that women with ovarian cancer have a very poor prognosis. If we can diagnose ovarian cancer early, that can actually change from, let's say, a 30% survival rate all the way up to 90% at stage 1. We're currently conducting with the University of Queensland a biomarker validation study. We're actually in the process of analysing that data, and we've said to the market that we expect to release that data before the end of the year. So we're looking forward to providing the results to that. We think this is a really exciting project because it actually validates the ExoNet technology, provides a fantastic solution for women to get early detection of ovarian cancer, and enables us to actually publish these results in market to support our ExoNet technology as well as our future diagnostics pipeline. This really says what I've already said, so I'll move on. Exosome therapeutics, so that's our new area. We've actually been working in the background on this for about the last 2 years. We developed a part of our technology we call ExoAce, and it's for actually capturing exosomes at scale direct from cell lines. So what we're really doing now is we are developing, we call them weaponized exosomes. We've engineered them so that they can target and kill cancer. Why have we done that? Essentially, they operate very similar to cell therapies, and everybody's heard of CAR T therapy out there, and CAR T therapy's done a remarkable job at improving patient outcomes for blood-based cancers. The way that cell therapies actually work is a lot of the activity of them comes from the exosomes that they release. So what we do is we actually take the exosomes from the CAR T, if you like, which is the engineered T cell or an engineered NK cell, and essentially we basically target them to cancer, and we don't get any of the negative safety issues surrounding CAR T therapies. So they become very efficacious therapies that are very safe for treatment of solid tumours, and that's what our focus is. So really this basically says that we're focused on CAR exosomes. We use our own technology, which is patented, to pull out those exosomes at scale. It's a really important thing because that was one of the big gaps in exosome therapeutics, was being able to actually isolate these exosomes at scale. So that's our own technology. We basically use different types of antibodies, we call them CARs, to target these to different cancers. Our first focus is actually on triple-negative breast cancer. And the reason we're focused on triple-negative breast cancer is it doesn't have any targeted therapies available for it today, so it's an unmet need. So essentially, that's our very first target for our pipeline. In terms of some of the benefits that are offered, we get improved efficacy. We can dose our exosome therapy multiple times, whereas with a CAR therapy, you can only do a single dose. And that dose costs about $300,000 US. So we expect, because we're actually taking our exosomes from what we call an immortalised natural killer cell line, we put a target on the surface of the immortalised NK cells. They then express these or release these exosomes. Those exosomes contain the same CAR target to go to the tumour, and we can treat multiple patients. So we've solved a whole lot of problems around manufacturing and logistics that current autologous CAR T therapies have, meaning that reduces the dose. We can basically provide a therapeutic to a patient within a week, massively reduced treatment costs, and an improved safety profile, because exosomes can't replicate in the body, so they can't cause secondary tumours. They also won't cause what's called cytokine release syndrome or immune rejection from the body. So we think that this is a very exciting pipeline. First target, triple-negative breast cancer. Potential other targets could be brain cancer because of the ability of exosomes to cross the blood-brain barrier, and there's many other opportunities. What we have done is we recently, in fact, our Professor Greg Rice there recently presented a poster presentation on our proof of concept data at a cell therapy conference in Copenhagen that was very well received. Basically, we showed that we could engineer our exosomes with an antibody to target them to breast cancer, that the exosomes released had that same antibody on the surface, rather important for them to do their job. that these exosomes were isolated by our technology called XOACE, and that we killed breast cancer in breast cancer cell lines. So that was our initial proof of concept data, of which we expect to release new data, in actual fact, before the end of the year on our NK cell line. So another area that you can expect to see shortly. In terms of where we're at, we've established what we call our master cell banks. We know our XOACE isolation technology does what it needs to do. We've released initial in vitro data, and we're in the middle of basically establishing contracts and various collaborations in order to fast track this exosome therapeutic technology towards the clinic, basically, which is the focus of the company. And we expect to move into in vivo studies, which is animal studies, early next year. The last area I just want to cover before finishing off is our sub-B2M technology. Everybody knows, I think by now, that technology basically is a protein. We license it from the University of Adelaide. We use it to target certain tumours that are only found in cancer cells. The technology helps to improve the sensitivity and specificity of existing cancer biomarker tests that are used for monitoring a range of different cancers. Our first focus has been on what we call the CA15-3 test, which is a breast cancer monitoring test. So that test is actually available on market by companies like Roche and Abbott. What we've done is shown that we've developed a better test, which includes our own technology, sub-B2M, as a reagent in that test. And as a result of using that technology, what we've done is we've improved the sensitivity and specificity over Roche's test, which was our comparator in this study. So we got 81% sensitivity and 93% specificity. So sensitivity means our ability to actually detect breast cancer, and specificity is about false positives. So when we say that we got 93% specificity, it means we only got 7% false positives, which means it's pretty good. But this is for monitoring, as I said, so that's after the patient's been diagnosed with disease. And effectively, we did a monitoring study afterwards, and we showed that the test was effective for monitoring, which is its purpose. In terms of our upcoming, I'll just move on to our upcoming catalysts. We did a different layout just so that you can focus on the upcoming quarters. You can see before the end of the year, as I've mentioned, you can expect a couple of new reports on data from both our exosome ovarian cancer biomarker validation study, which should be pretty near term, and also our in vitro proof of concept work on our natural killer therapeutic. So we will be presenting that soon. In the next quarter, we've got a lot of work going on around clients that we're working with Promega for exosome diagnostics, and that's really what the exosome diagnostic agreement's around. Regarding an upcoming collaboration, we should be producing some more data around our CAR-T exosomes. And then moving throughout the next sort of 12 months, we'll expect to produce animal data, which is really important data when you're developing exosome therapeutics, which shows that it's safe and effective in mouse models. So we'll be able to do that both with our T-cell-based exosome therapeutics and our NK. On the diagnostic side, we've got a lot going on for both our sub-B2M diagnostics and our exosome diagnostics. And importantly, we expect to see revenue growth across our exosome research tools. And that actually includes both ExoNet and NeuroNet. I think, David, ready to hand over to you?
Yeah, no, stay up there. I'd open it up for questions, but I thought coming closer, and I thought I might just ask you a few questions myself, and then open it up if there's others. As I said right at the start, I just want to sort of dumb it down a bit. I'm not sure exactly where to start, but let's just start with this concept of capturing exosomes, because we tend to talk about capturing it for cancer, breast cancer, let's say, messages. So I'm thinking about this whole capturing thing as you're painting me a picture where there's a cell and the cell gives off signals, and you can read those signals.
Mhm.
To what extent is that capturing versus identifying? When you then moved on to neuro messaging and so on and so forth, I'm looking at the platform as being a means of identifying as opposed to capturing. The information is coming anyway, isn't it? So am I being pedantic or?
Essentially what our technology does is it does capture. You can't do anything with an exosome if you don't capture it first. So firstly, we capture it. That then enables us to look inside and identify the biomarkers. So we get to do both. And our technology, the way we use it, and particularly when we use it with Promega's extraction technologies for RNA, it actually enables us first to capture the exosome, then to look inside and extract out the RNA, and then we basically turn that into a diagnostic.
So just the hardest example I had to get around my head was you were talking about messaging where there's been a rejection of tissue or rejection of a limb or something like that. That's a completely different message, obviously, to a cancer messaging.
Yeah.
But what, again, are we looking for, how are we building that logic up? Do we go and look for a database of people who have had, let's say, limb rejection and the sorts of blood tests that they have, and then we analyse that and we see whether there's any common markers? That's the sort of...
So whenever we develop new diagnostics or our collaborators develop new diagnostics using our technology, what they do is they take what's called cases, which means people with disease, and controls, which means healthy people. They take the exosomes from both the healthies and the diseased, and then we have a look inside to see what's different between the cases and the controls. The bit that's different is actually what we use, whether it's for cancer patients or whether it's for limb rejection, because it's whatever's different tells us whether that person has cancer or whether they're likely to reject a transplant.
And given a lot of these databases are public, public databases, blood samples and so on and so forth, so how hard is it to make sure we've got intellectual property in being able to just identify? That's the easy part, it seems to me, but what's our IP position on that? Or are we using, is it first in class, as it were, and then to use that as a therapeutic? Is that where you're heading to? Or?
So the company's got a broad range of intellectual property around its exosome capture technologies, its diagnostics and its therapeutics. So from an exosome capture technology, that covers exactly what our technology is, such as a magnet with a matrix around it, different number of types of antibodies to enable us to pull out different types of exosomes from the body. That's a really important intellectual property, or let's just call it patent portfolio for our capture technology. Then we've got around our diagnostics, that's when we actually look at the biomarkers and we patent the combination of biomarkers and the algorithm that enables us to detect disease and determine whether someone has cancer or not. Our partners, for example, or sorry, I call them customers in actual fact, of ExoNet or NeuroNet, they can actually develop their own biomarker combinations and patent that. But the way that we would work with them in the future is we could license our technology to them for their exosome diagnostic, and we would also sell them the product to use in their exosome diagnostic kit.
So let's go specifically to, let's say, the University of Queensland and the ovarian cancer testing project that we've got going with them. And as you quite rightly said, we'll probably have that out, the data and the success or otherwise of that within the week, I imagine. But I'm interested in how we get to a commercialisation. So let's say, for example, that the project we've got with University of Queensland, which I imagine is using databases of other people's blood that's already been collected, right? So we're just doing this lookup and what's different and what's not.
They've actually discovered, so part of what they did was, how I said, we designed experiments. They took cases of ovarian cancer, healthy people, they extracted the exosomes using our technology. They discovered different biomarkers in those in that technology. They do a pattern around those combinations of biomarkers. So that's actually, that bit's the University of Queensland's IP. We have a license then to use that, to commercialise that IP.
Okay, so let's say it's all blue sky and green lights and so forth. And there is no reliable test at the moment for ovarian cancer. What's to stop us? What are the steps between those results and us walking along the road to Gribbles and going, listen, here's a test. Any women you get in, given this additional test on their ovarian. What's the steps between that and commercialisation?
There's quite a lot of steps in developing a diagnostic. Essentially, we have to first first we have to validate the biomarkers. That's our upcoming study results. So that's just about done. The next step is what we call analytical validation. Analytical validation is where we prove that the test is robust, so it doesn't change each time we do it. It's reproducible, whether it's in one lab or another lab, on an instrument platform. So that's quite a big job, and that's one of our upcoming jobs. Once we've done that, then we clinically validate in forward-looking patient populations, and we need to discuss with regulatory authorities what the numbers look like. So that's one of the upcoming jobs will be to work with the FDA, for example, around what patient numbers they expect us to have. One of the opportunities for our exosome ovarian cancer test is because it's for screening and there's such an unmet need, it could potentially get what's called breakthrough device designation, which enables us to speed the path to market costs, and that's what we'll be seeking to achieve with this test.
Okay, I think I understand all that. Let me just turn to Promega for a moment. And as you quite rightly said, they've currently got, in the space of 5 months, 35 or thereabouts customers spread across clinics and academia and so forth. I love the fact that we're a platform and that there's all different types of uses being investigated, some of which we probably never thought of before, increasingly so. But to what extent are we sharing information with Promega and getting involved in, or certainly staying close to whatever these researchers are using it for?
Sure. So we basically have weekly meetings with the Promega team on the sales side. We have 2 weekly to monthly meetings on research and development. So there's 2 focuses. One is the commercial and the applications, as you said. We have a business development person in the US who works very closely with Promega on a day-to-day basis. And that's really to provide technical support to customers and to understand the applications. Sometimes when we're working with customers, they want more information about different things. That information is fed back to us. Our R&D team will usually respond if Promega doesn't already know the answer. Frankly, they're pretty damn good now and they know most of the answers. And importantly, Promega's doing a huge amount of research and development around our product, ExoNet, and combining them with their product to give customers complete solutions and that enable their research. So we do this, we've got 3 ongoing projects with them at the moment. So there's a lot of work going on between the 2 companies.
So is Promega all things to us though? I mean, is there any reason why, well, first of all, do we have an exclusive relationship with them? And if not, who else should we be talking to apart from Promega to broaden our reach, if you like?
Sure. So the agreement with Promega is a non-exclusive global distribution agreement. So they can distribute our product globally, which they are doing. They've got branch offices all over the globe, as well as distributors that they use. They've actually been selling this product now in the US, Europe, and Asia. They're also trying to expand into the Middle East with sales. They have a head office here, not head office, a branch office here in Australia. And actually just appointed a new GM who's very focused on understanding ExoNet. In terms of what we do with direct customers, we tend to work directly with the diagnostic companies and the therapeutic companies themselves. And the reason being is sometimes they want to get a custom product, which means that we can then pull out a certain exosome subset that gives them a better test, which is what they're seeking. So we tend to do those types of relationships direct. We could expand to other distributors, but we think that Promega is investing heavily into supporting this technology. It'll just take some time to build the sales pipeline, but they're definitely very focused. And they recently actually did a sales training at the head office in Madison, where they had all their staff over a couple of weeks come out from all around the globe. And that included training on ExoNet, which our business development person in the US delivered with the Promega staff.
So whatever our relationship with them is, it doesn't stop us from going and doing a deal directly with the University of Queensland.
No, absolutely not.
And are we making sure we keep Promega informed about what we're doing so that they can use that when they go to sell one of the universities?
We absolutely do. Yep. And one of the things we're doing, so whenever we have our calls with them, they provide an update on their activities, we provide an update on ours, and jointly, some of these important opportunities like our exosome ovarian cancer test will turn into profiles that we can use to promote the products to a broader range of customers.
Yeah. Okay, Jen, finally, you put up a sort of a timeline there, but just to dumb it down for everybody, when you're sitting here thinking about the future of IIQ and how our share price is going to get to $1, what are your sort of two or three focus points for how we should be looking at it?
Sure. So our exosome ovarian cancer test data coming up, we consider to be really important data in terms of developing an exosome-based test. Not only does it support our ExoNet technology and therefore sales of that technology, it also supports an actual real test being developed for screening. So that's important. That enables us to advance into the next stages of development. So that's one. The other is the fact that we're moving fast-tracking our exosome therapeutics pipeline, and we expect to be able to produce animal data next year. That in any therapeutics company is a really important catalyst, and we expect to have that data next year. So that we're very much looking forward to producing. And then obviously commercial growth of our ExoNet technologies and various partnerships that we expect to be able to talk about next year.
And you touched briefly on regulatory and, do we have a dialogue going on with any regulators at the moment around the world about exonets and where we're taking it, what might be coming?
So our diagnostic technologies are at the stage that we are about to commence discussions with regulatory authorities, particularly in the US, so with the FDA.
Yeah. And do we have a consultant who will guide us on that, or where are we at in terms of?
Yep. So we're talking with a consultant around our regulatory strategy and how we approach the US. Yes.
Okay, great. I'm going to get Greg up in a moment, but before I do, is there any questions on the floor that would like to ask Leanne? Not to stop you, but yes.
My name's Jim Perry. I think you're doing very exciting work. For your results. I was just concerned, I'm not sure if you're concerned. The company basically is an R&D company, but it appears you're having only approximately half on R&D development construction.
So basically the companies clearly require a team to move anything forward. And part of that team obviously support around finance and admin, at corporate costs. We spend a lot of money on R&D, but because it's early stage R&D, it's not as we, let's say, advance our pipeline, R&D expenses start to go up. That actually means, of course, that we need more money in order to fund that R&D. But I think the company's pretty well balanced and conservative with the way it spends its money. Compared to a lot of biotech companies, I think we manage our money extremely well.
Mark, do you have any questions?
Yeah, we do have a handful of questions. Can you give an approximation of near to midterm future exonet sales, whether in dollar terms or growth rates?
Well, I'll answer that because, we're not really yet giving forecasts and so forth. I think Leanne's given as much as we need in terms of what our current relationship with Promega and with Teddy and they're doing on the side. But we're both, I think, Promega and ourselves, still sucking and seeing it as it goes, I think.
Okay. Another question on exosomes. How does our exosome tech compare with that of other leading exosome tech companies?
Well, we might let Greg answer this. But yeah, you can stay there and come up.
Yeah, look, a technology differs from other companies. The prime and important feature is that we are able to specifically capture subpopulations of exosomes. So ones that come from the liver, ones that come from the heart, ones that comes from breast cancer, for example. The other technologies appear to be more general, so they capture a broad range. So the issue is that when you look for biomarkers in broad range populations, you've got signal to noise ratio effects, and it's harder to actually develop diagnostics using that sort of technology. So it's really about this capacity to capture that Leon talked about, and that distinguishes our technology from other companies.
Good.
Okay, yeah, a couple more questions, this time on sub-B2M breast cancer. The in-market date seemed to have been pushed out from a couple of years ago. How confident are we in the current in-market date of Q4 2025 for product release?
Basically, as we develop the test and we talk with potential partners of that test, so giving an example what a potential partner for the test is, that's essentially large clinical laboratories. They could be Quest Diagnostics, LabCorp, Sonic Healthcare. They tell us what they want to see before they'll take the test on. They then have to analytically and clinically validate the test in their lab. It's actually a relatively short process. They just repeat quickly what we did, but they want us to do the work first. So, for example, one of the things that we're doing at the moment is showing disease specificity of our sub-B2M breast cancer test. We're transferring it to a bead-based assay so it can be used on automated instruments that are used in these big labs, and that will take some time. But we are focused on commercialising, getting a commercial partner and getting that test into the market in the second half of 2025.
Thanks, Leon. One further question is, where are we at with securing our laboratory partner for our breast cancer test too?
I think I just answered that one.
Yep.
Okay, Greg, I might ask you to come up and just. So, Greg, well, first and foremost, here's some living proof, walking proof that we're spending some money on R&D. Greg, this whole thing about capture we get, now identification I get, just let me test you though, I give you a vial of blood. I don't tell you where it's come from.
Yep.
Do you know where it's come from in the body? Can you tell when you look at it that this is from the liver or from?
Yeah, look, this is really the underlying benefits of our technology. And it comes back to this issue that Leon talked about with capture and the difference between our technology and other technologies. Just to elaborate a little bit more on the other technologies that are available, most of those technologies isolate exosomes based on their physical characteristics. So how big they are, how much do they weigh, what's their electrical charge, or they isolate them on common features of exosomes.
Yeah.
So there is no way using those sort of technologies that you can pull out a specific set of exosomes from lung, or from the liver, or from the breast.
Yeah.
Our technology actually targets markers or features of exosomes that are characteristic of and only come from, for example, breast cancer cells or lung cancer cells.
Yeah.
So that's the difference in the technology. So if you gave me a vial of blood and we had the right exonet to target a particular tissue, we could say whether there were exosomes from that particular tissue in that vial.
Okay, I got it. So, all right, let me take it another step further then, because I think most people will understand the difference between diagnostic and therapeutic. Okay, we've got the blood, we know where it came from, we identify the markers. Okay, there's cancer in that breast. Fine. What Leanne described you as doing is putting a bomb in it and sending it back.
Yeah.
So, okay, let's just separate those two things. First of all, how do I actually send it back? I know where it's come from,
okay,
but how do I send it back? I can't diagnose it. You know what I mean?
Sure. Okay. But the easy way to explain it, I use an analogy of the Australian parcel post. If I want to send you a set of Riddell wine glasses,
Please.
what I do, absolutely. What I do is I wrap those glasses up in some protective material, bubble wrap. I put it in a sturdy box so I can protect it in transit. I stick an address label on it.
Yeah.
My sender, a label on it as well.
Yeah.
Give it to Australia Post. They deliver it to you. You open it. The Riddell glasses are intact because it's been protected in transit, and you can enjoy your Barolo in a suitable glass. Cells do exactly the same. When they have an important message that they want to send to another cell, they wrap it in a protective package. That's your exosome.
Yeah.
And they're able to address it on the surface so that when they actually release it into the blood, it will go to that particular cell and deliver the message.
Yeah.
Our technology allows us to intercept the mail.
Yeah.
Read the mail. That's the that's the diagnostic side of our business.
Yeah. Yeah.
We understand where it comes from and what the message is. And the message might be that I'm a normal cell, I'm fine. Or it could be that I'm a cancer cell, and that tells us about what we can do. The other thing that we can do is that we can then make those exosomes into killer exosomes.
Yeah.
And put them back with that address label on, so they actually get to the cell.
Yeah. That's the bit I'm asking
Yeah.
about. How do you get it back into the postman's hand?
As Leo mentioned, we have technologies for isolating exosomes for therapeutic use.
Yeah.
So basically, we're putting an address label on the outside of those exosomes.
Yeah.
And then infusing them back into people.
Yeah. Okay, it's that bit I'm having trouble with. Is how do I get that address label on it? You know, I mean, it's not as simple as just saying send to the breast.
Well, it is actually.
Is it?
Yeah. Because we know, for example, breast cancer cells express on their surface specific proteins that are associated with them being cancer cells.
Yeah.
So what we do is
So you attach sugars to it or something like that.
well, we put on the outside of the exosome an antibody against that particular protein.
Yeah.
So that it will then specifically target.
Yeah.
So, for example, one of the common breast cancers is called an EGFR positive. So there's a protein on the actual cancer itself, EGFR, that we can target. And it's not expressed by other cells.
Yeah.
So that way we can actually address it and we can target it.
Yeah. All right, that's good. Is there any questions for Greg on the floor? And nothing online that you haven't already answered. Well, look, thank you for that, Greg.